do my best. So, first slide, please. There we go. So I want to thank Dr. Carrero and Dr. Leuenberg for inviting me to speak today. It's a pleasure to be here. Thank you. I'm from Toronto, and this is York University, 55,000 undergraduate students, and the home of the Muscle Health Research Center, the biggest muscle health center in Canada, potentially in North America, 25 faculty members, 120 trainees. If you're interested in muscle health, and you want to come to North America, this is the place to be, okay? Toronto, no question about it. I'm very impressed by Italian science in muscle and muscle biology. It's so impressive to me. I congratulate you as a community of scientists in muscle biology and muscle health. I'm in awe of the work that you're doing here in Italy. So let's see if we can move the slides. So, yeah, okay. No, I prefer that one, okay? Yeah. So I'm going to talk about mitochondria and lysosomes. Let me see how I'm doing. Okay. So you all are well aware of mitochondrial plasticity, right? Mitochondrial plasticity in muscle, I don't have to tell you this really, because it's very well known that with, here's our mitochondria in healthy muscle. You've got a few of them here. With regular exercise, of course, you increase the mitochondrial content, subsarcolemmal, intermyofibrillar mitochondria, and we have a more greater muscle health. With age and disuse, of course, one of the prominent features is a loss of mitochondria and muscle atrophy. And I can make a case for you that that mitochondrial dysfunction, when it's going wrong, contributes meaningfully to the atrophy of muscle and the sarcopenia that occurs during aging. And of course, mitochondria are directly related to cardiorespiratory health, right, and your longevity. So it's in our interests to maintain and understand what's going on with mitochondrial health during conditions of disuse, which, you know, inactivity is a form of disuse, and of course with age, where we have a decrease in mitochondrial content. So that's been a focus for me for a number of years. I take a slide from one of my colleagues, Tarnopolsky, who works down the road from me at McMaster University, just to show some human data on mitochondrial content. Here is a young muscle, here's an older muscle. It's not a great EM, but mitochondria are bigger in young muscle. They have more of a reticulum in old muscle. Of course, it's more fragmented, smaller mitochondria. There's irregular mitochondria as well. If you do a count of mitochondria, I don't know how he did that, but anyway, if you look at the count of mitochondria, it's reduced. If you look at mitochondrial markers, of course, it's all reduced. And the mitochondria are fragmented. And the more fragmented they are, the more dysfunctional they are. So we need to understand how this happens. So how this happens is, the way I study this is I try to understand the mitochondrial life cycle. So the life cycle consists of whatever signals are impinging on the nucleus here to drive the synthesis or biogenesis of mitochondria. Here's healthy muscle mitochondria existing in a reticulum, a network, right? That's how it exists in skeletal muscle, as you know. PGC1 is a driver, of course, of nuclear genes encoding mitochondrial proteins. Then we fuse these smaller pieces together to make the reticulum. And when there's a portion of the mitochondrion that becomes dysfunctional here, that portion undergoes fission through regulatory proteins, and these fragments of mitochondria are dysfunctional. They exhibit a reduced membrane potential. They have high reactive oxygen species. Time to get rid of them. So we tag those mitochondria with proteins in the process of mitophagy. This is the degradation side of things. Right, we have LC3P62 that tags these mitochondria. We transport them on microtubules to the lysosome where mitophagy takes place, and we degrade these mitochondria. And in order to study this process, you have to measure the flux rate, which means you have to inhibit the pathway and see how fast these mitochondria are degraded in the presence and absence of an inhibitor. If you don't measure flux, you'll never publish anything in this field. So it's very important to measure flux. And I'm going to make the case for you that with aging muscle, there's a greater mitophagy rate than there is biogenesis. We know that with age there's impairments in mitochondrial biogenesis that leads to less high quality mitochondria, and we also know that mitophagy is high. I'm going to show you the data on the next page that shows you that the degradation rate of mitochondria in aged muscle is high. So what does that mean? It means biogenesis is lower than mitophagy and the mitochondrial content will go down, right, in aged muscle, and that's what we see. I'm also going to tell you a story about TFEB and TFE3. These are two transcription factors that regulate mitophagy, supposed to regulate mitophagy. TFEB, of course, very famous, very prominent, and TFE3 less so, and so we naturally picked on the underdog to study. We decided we were going to study TFE3 quite a bit. I'm going to tell you that mitophagy is high in aged muscle, but if you improve the quality of the mitochondria with exercise, then the mitophagy rate is reduced because you don't need to break down good quality mitochondria. You can keep them. You don't need to signal the, an increase in mitophagy. Let me see. Yeah. So that's what these data show here. This was done by Heather Carter, published in 2018, where we looked at the tagging of mitochondria and compared young and aged mitochondria using inhibitors, plus or minus an inhibitor to measure flux, and we saw that the aged muscle had a higher mitophagy rate. But if we use a chronic stimulation, an implantable, this is on the back of the animal, but we more recently implanted stimulators that Dr. Jarvis made for us and put them inside the animal and chronically stimulate one side of the leg. We, like a chronic training model on one leg, we see that the flux is reduced in young animals and also reduced in the aged animals because we're producing better quality mitochondria with chronic exercise. Okay, so the higher mitophagy flux that we see with age contributes to the reduced mitochondrial content in aged muscle, but it's attenuated by chronic exercise because of that improved quality of mitochondria that you produce with exercise. So here is what we think is happening between age when you compare young muscle with aged muscle. We have a normal rate of biogenesis. We're producing average quality mitochondria. We take some dysfunctional mitochondria and direct them to lysosomes. We have a normal lysosomal degradation rate, normal lysosome number. Everything's great, fantastic. With aged muscle, we have poor, less good quality biogenesis, in part PGC1 related. We've shown that before, so we have poor quality mitochondria. That means we have to direct more of these mitochondria to an autophagosome and direct more of them to the lysosome. That's what the thick arrow means. We've got more mitophagy flux happening with age. Problem is right here is my feeling. Right here is the problem. We have a good flow to the lysosome, but if you look at electron microscope pictures of muscle and look at the lysosomes, you see a lot of lipofuscin. So a lot of lipofuscin is an indicator of poor lysosomal function, right? Nobody's ever isolated lysosomes from muscle and actually measured the function, and that's what we're trying to do, but this is an indirect indicator that this is a problem here. And the more lysosome dysfunction you have, the more lysosomes are generated to try to compensate for that. So there's an overexpression of a lot of lysosomal proteins, and I'll show you that data. So and the other thing we didn't know about is we don't know anything about the effects of biological sex on this process, right? And the effects of exercise. So we took some mice, we had young mice and aged mice, and they were divided into male and females. We don't have an overwhelming number here, but anyway, we're able to get some statistical differences. Young and aged animals, and we exercised them, and we looked at various markers of mitophagy and respiration. So I've got a complicated slide here, but let me tell you what happens if you don't separate males and females, first of all. If you just have young and old animals, you just say, okay, let's analyze. Okay, so here's complex 4. This is a mitochondrial protein. If you compare males and females with age, you can see combined males and females, you see a little bit of a reduction with age, but it's obscured. You're obscuring the sex difference if you don't separate them. So here, if you look at complex 4 protein in males with age, it goes down by 45%. In females, there is no reduction. This is young, this is old, no reduction. Here's another one, complex 3, goes down in males, no change in females. Now, if you look at the respiration rate, so you have young male, young female, old female mouse. So no change in content in the female, but a drop in function, okay? So females do not reduce their mitochondrial content with age, but they reduce the function. Yeah. So the question is, if that's a functional thing, does that trigger mitophagy or lysosome synthesis? So we wanted to address that. No. Yeah. So using transgenic mitochondria mice, we can show that another measure of mitophagy flux is elevated with age. So this is shown here. That's just additional data. But if you look at the markers of mitophagy like Parkin and BNIP3, if you look at the difference between males and females, here's a young male and a young female. Higher Parkin, no change in BNIP3. Look at how big these go up with age. Huge increases in these mitophagy proteins in both males and females, but in particular in female mice. Huge changes. Same thing with lysosomal proteins. Lysosomes are higher in females compared to males. Big increase with age. Lysosomes are higher in females compared to males. Big increase with age, especially in the males to catch up to the females. So females have more lysosomes than males, at least in the mouse. So then we looked at the regulators of mitophagy, both TFEB and TFE3. These are the transcription factors that respond to exercise, go to the nucleus and synthesize lysosomal proteins and autophagy proteins, the regulators. So here again, TFEB, very important, higher in females. TFE3, the second most important one, higher in females. Big increase with age in both males and females. So the direction toward making lysosomes is very much elevated with age to compensate for that dysfunction that I showed you with the lipofuscin earlier. So we're making a lot of lysosomes with age. Whether they work or not is the question. So in combination with Dr. Leuenberg and Anna Pica, we did some preliminary work to just look at that in human subjects. We had old participants, 14 of them. They were about 78 years old. And I have to say, I still think we need to do more work on this. We see some, this is the elevation here in P62 and BNIP3 indicating elevated autophagy, potentially elevated mitophagy. We saw a lot of variability in the TFEB and markers of lysosomes in older subjects, but we did not separate them according to sex, and we have to do that. And once we do that, we'll have a more clear picture of what's going on in humans. You can see the tremendous variability that we have here, and we need to separate out. And this is, again, what happens when you don't separate them. You obscure potential sex differences between males and females. So we have more work to do in that area. Does acute exercise move these proteins to the nucleus? In the mouse, you know, exercise increases calcium, that activates TFEB to go to the nucleus. We have our running animal again, and we look at the percent of nuclear TFEB. So in young animals, in males, we get translocation of TFEB with exercise, acute exercise animal, going to the nucleus. TFEB moving to the nucleus, same in females, going to the nucleus. With age, we have no translocation of TFEB. At least we couldn't detect translocation of TFEB. So no change between non-exercised and exercised, no change, in fact. Okay, so something going on with translocation of TFEB in age, we're unable to see that. However, we can activate TFEB transcription with exercise. In the young animal, we see when we use a reporter assay and inject the muscle into the animal, we can see activation of TFEB promoter in young animals, and the same activation although it's depressed, transcription seems to be depressed in the aged animal, but it's still activatable by exercise, which is good because exercise can be therapeutic in helping get TFEB into the nucleus, whether you're young or whether you're old. It doesn't matter. But somehow we have to figure out why that's not translocating into the nucleus. Have you looked at lysosomes lately? When you think about muscle biology, do you think about lysosomes? I never did. Here's the lysosomes here. Did you ever think there were that many lysosomes in a muscle fiber? Of course, here's the nucleus. Here's our mitochondria, all in red. Huge numbers of lysosomes inside muscle. I can watch them move. I won't show you the movie in the interest of time, but we can look at lysosome dynamics as we've looked at mitochondrial dynamics in the past to see how the kinetics of those and how they move. Oh, there, look, it's on anyway. Okay, so to summarize just a little bit, males and females, males have less mitochondria than females, and that holds up in humans and in mice, okay? Females have more lysosomes than males. Females seem to direct more autophagosomes to the lysosomes, so mitophagy is greater in females. As we get older, as the mice get older, male mice lose mitochondrial content with age. Females do not, but they have more dysfunctional mitochondria. We have an increase in lysosomes in both males and in females. We don't know whether they're functional or not. That remains to be decided. TFEB goes into the nucleus with exercise in females, a little bit more in males it seems, but we can translocate TFEB to the nucleus in males and females when they're young. It doesn't seem to be happening with age, but we can activate TFEB promoter activity equally with exercise in both young and old, and I think males and females, although I need to do more work on that sex difference. So exercise definitely therapeutic, and we have to work on what's going on with age. So the two proteins that I've been talking about, well, mainly I've been talking about TFEB. The underdog in this world is TFE3. TFE3 regulates metabolism in cooperation with TFEB. They're both, there's a list of papers here that shows the cooperation between TFEB and TFE3 in regulating metabolism, regulating lysosomes, regulating autophagy. There's quite a bit of information there, particularly on TFEB. So naturally, we wanted to look at TFE3 and see what's going on with exercise training, first of all, if you don't have TFE3, and then with denervation or disuse if you don't have TFE3. So we used a knockout animal. This is a whole body knockout animal. We put them on running wheels, and we looked at the distance they ran over 6 weeks. The knockouts and the wild types ran 200 kilometers each. That's fine. And we looked at the fatigue response over time. The wild type animals, if you stimulate the muscle in situ, make the muscle contract and look at the fatigue rate. With training, you get an improvement in performance. This is the wild type animal. Improved performance, less fatigue. You don't see that in the knockout. We do not see an improvement in fatigability in the knockout animal. And the reason for that is because in the wild type we can increase mitochondrial content. That's not significant in the knockout. We can increase respiration in the wild type. It's not doing anything like that in the knockout. And the percent changes are shown over here. Get a bigger increase in mitochondrial content in the wild type. Fatigue resistance improves, respiration improves. These things are not happening in the knockout animal. We have higher ROS production in the knockout animal as well compared to the wild type. So just based on this, and this is still unpublished, we believe that TFE3 has more of a role in regulating biogenesis in response to exercise than it does in mitophagy, because I'm going to show you what happens here to mitophagy, and surprisingly enough, TFE3 is doing virtually nothing when you knock it out in terms of mitophagy. So we took the TFE3 knockout in wild-type mice, we denervated them for 7 days, and we looked at males and females to see what happens to mitochondria, what happens to mitophagy. Here's a simple graph that shows the mitochondrial content. And similar to the aging phenomenon, what we see here in males, this is wild-type males with 7 days of denervation. Everybody knows what happens with denervation. Mitochondrial content goes down, okay? It's like disuse, it goes down. Yes, it goes down in males, but it does not change in females. These are wild-type animals. No change. In the knockout, it goes down in males. In the females, it goes up. There's something weird going on there. So what about the function? So females are behaving differently. And if you don't separate them according to sex, you're going to obscure the real differences in those sex differences. So with respiration, again, I've got the 4 groups here: wild-type male, wild-type female, knockout male, knockout female. I'll just summarize what's going on here. With denervation in the black bar, you generally see a decrease in respiration across all the groups. You see an increase in ROS emission across all the groups. So this is increasing mitochondrial dysfunction with denervation. But interestingly, the mitophagy flux that could be triggered by this mitochondrial dysfunction really was not activated. But one of the things that was prominent was mitophagy flux is elevated in females compared to males, whether you have TFE3 or you do not. Okay? So females get rid of dysfunctional mitochondria more effectively than males do. Mitophagy flux is elevated. I'll skip the Parkin data in the interest of time. What about the lysosomal markers, lysosomal proteins? So here again, cathepsin D, the mature form of cathepsin D, is higher in females, goes up dramatically with denervation. As all lysosomal proteins do. This is a great model if you want to study the increase in lysosomes. Cut the nerve to the muscle and you'll see a marked elevation in all kinds of lysosomal proteins like cathepsin D. There's no difference between whether you have TFE3 or whether you do not. So TFE3 is not regulating that in any way. So to summarize that little bit, females display more mitochondrial protein with age and with denervation compared to males, but show evidence of dysfunction of mitochondria with age. They have higher levels of lysosomes and regulatory factors, TFEB and TFE3, with age and in response to denervation, and they have elevated mitophagy markers and flux. Lack of TFE3 impairs biogenesis of mitochondria, but it does not seem to impair what it's supposed to be doing, which is mitophagy, lysosome synthesis, or mitochondrial function in males or females, and that was a surprise to us. So then we wanted to look and see how these two cooperate, TFEB and TFE3. So we went to cell culture. These are myotubes, and we knocked down TFE3 and TFEB either together or individually using siRNA. So we look at TFEB, you can knock down TFEB. You look at TFE3, we can knock down TFE3, and we don't get compensation in the other one, okay? So we're able to knock them down individually in these myotubes. And if we look at mitophagy flux, this is the individual knockout, this is the double knockout where we knocked out both of them. Here's basally, this is not in response to contractile activity or exercise. P62 mitophagy flux is not different. Not different. This is whether you knock down TFE3 or TFEB, it hasn't changed. Doesn't change when you knock down one or the other individually. But when you knock them both down together, this is what we see with double knockout. We see a decrease in mitophagy flux here based on LC3 tagging or P62 tagging. So when you have them both knocked out, then you have an impact, but not individually because they obviously the presence of the other one clearly compensates for the one that's missing. If we use this stimulation model here, you can see these myotubes contracting. There's my ancient stimulator that I built in 1999, and we've been using this to stimulate myotubes for several decades now. We can make them contract in 6-well dishes, and that's our exercise in a dish model, right? And we look to see if contractile activity can rescue some of this. So we have the double knockout animals, and we have control and what we call CCA, or chronic contractile activity. So with chronic contractile activity, you look at lysosomes. We've shown this in vivo in animals too. When you chronically stimulate the muscle, you get an increase in lysosomes. So exercise stimulates lysosomal biogenesis. And the question is, you can measure markers of lysosomes, but whether they're functional or not is the thing that we still need to confirm. We've done it here. This is an increase in lysosomal area with chronic contractile activity. It is unimpeded by the knockout of both TFEB and TFE3, okay? No impact. However, the function, this is a marker, DQBSA, which is a marker, one of the only markers. You can see the green puncto over here. Maybe you can see it with this DQBSA fluoresces when you have functional lysosomes. And we see that with the double knockout, you see under control conditions, you have a reduction in lysosomal function. No change in content, but reduction in mitochondrial function. Exercise improves lysosomal function by itself. So we got, in this model, we have an increase in lysosomal function with contractile activity. And that's shown also here with this cathepsin B ratio of the precursor cathepsin converted to the mature cathepsin. Okay, contractile activity accelerates the conversion of the precursor into the mature form, and you can see that modest, very modest here, but the double knockout really impairs that. So lysosomal function is definitely impaired when you don't have TFEB or TFE3, TFEB and TFE3 together. So the final few words I want to say is we're interested in what, TFEB and TFE3 are obviously important in biogenesis and lysosomal function, et cetera. What is it that controls TFEB and TFE3? What is it? So we looked upstream. I don't know why I'm interested in transcription factors all the time. I'm a control freak, I guess. But I like to understand what's controlling the next thing. So we looked at activating transcription factor 4 and its role because it responds to stress, right? It's the integrated stress response protein that regulates UPR, the unfolded protein response, mitochondrial UPR, et cetera. So we looked upstream and to see whether exercise actually had an impact as another stressor that would impact ATF4. And the great work by Chris Adams showed that it has a role in muscle atrophy. So that was one of the things that got us started. And if we look at one of his papers here, ATF4 is a negative regulator of muscle mass. If you knock out ATF4, muscle mass goes up. If you knock out ATF4, muscle mass improves in an aged animal. So you're attenuating the sarcopenia if you take away ATF4. Here, look, an increase in grip strength, an increase in specific force with a knockout of ATF4. So that data has nothing to do with mitochondria and has nothing to do with lysosomes. So we used a lentiviral overexpression or knockout in cells, and we looked at what happens with TFEB and TFE3. And when you overexpress ATF4, you get this big increase in TFE3, you get a big increase in TFEB. You knock out ATF4, you get a drop in TFE3, a drop in TFEB. So that was interesting. That was compelling for us. ATF4 regulates these transcription factors that we've been talking about. So if you look at mitochondrial expression, especially of things like PGC1, if you overexpress ATF4, PGC1 goes down. If you knock out ATF4, PGC1 goes up. Knocking out ATF4 reduces mitochondrial content. ATF4 is negatively regulating mitochondrial content. Here's what happens to lysosomes. Overexpress ATF4, bang, up go the lysosome. Knock down ATF4, boom, down go the lysosomes. So ATF4 is negatively regulating mitochondria, positively regulating lysosomes. Here's the final slide. Look what happens to ATF4 with age. Huge increase in ATF4 in muscle, liver, other tissues. ATF4 is promoting muscle atrophy, promoting a reduction in mitochondrial content. My hypothesis is this. ATF4 is a master regulator of the sarcopenic phenotype with age. There's where some work needs to be done, okay? Because it's producing the atrophy and it's knocking down mitochondrial content. And the mitochondrial content's going down because we have more lysosomes and less TFEB and TFE3. These are the people that were specifically involved in the work. Matt Triolo is Italian. Jonathan Memme is Italian heritage. She is from Portugal, and she's Oriental. TFE3, ATF4, sex and age. Thank you for your time and attention.